Good morning, everyone. My name is Pemba Sherpa, and this summer I had the opportunity to intern at the Shipman lab. During my time here, I worked with my mentors, Sierra and Rebecca, to evaluate purity of mitochondrial isolation methods. So for introduction, mitochondrial DNA mutations can cause mitochondrial diseases such as MELAS. We could fix these mutations with gene editing tools such as CRISPR-Cas9, which uses RNA as a guide. However, since the import and export of RNA into the mitochondria is not well known, we cannot send these RNAs into the mitochondria. The bigger picture of this project is to optimize a protocol to deliver RNA into the mitochondria. And for this, we need a pure mitochondria to avoid RNA contaminations from other organelles and to see if our RNA import went into the mitochondria. My part in this project was to isolate mitochondria using differential centrifugation, ultracentrifugation, and antibody isolation. And the goal was to optimize a Western blot protocol and to assess the purity of mitochondrial isolation methods. So for methods, in figure A, we have an image of the steps involved in mitochondrial isolation with differential centrifugation. The HEC293 T cells were harvested and then homogenized using a Downs homogenizer, and the mitochondria was then isolated using steps of low spin and high spin centrifugation. The mitochondrial pellets isolated from differential centrifugation were then used for BCA analysis to find protein concentrations and were then used as sample to perform Western blots. In figure B, we collected crude mitochondrial pellet from differential centrifugation and then diluted them in 20% percol solution. We used a syringe to carefully create a gradient of different percol concentration and then put them in the ultracentrifugation machine. The mitochondrial layer seen here was extracted using a pipette, and the pellet was then used as a sample for Western blot. For antibody-based isolation, we used a pipette to put the antibodies into the crude mitochondria isolate from differential centrifugation, which would then bind into the mitochondrial proteins and isolate mitochondria. I was able to use antibody samples from my mentor, which she had previously collected, and then perform Western blots on them. So here are the results from my Western blots. I loaded whole cell samples and mitochondrial samples into the gel. I used 8 different antibodies for different organelles, and the mitochondrial proteins are highlighted in pink. The presence of citrate synthase, porin, and TOM40 in the mitochondrial isolate samples confirms that mitochondrials were captured during differential centrifugation. However, we can also see contamination in the lysosome, ER, and the cytoplasm. Next, I have Western blots from my ultracentrifugation. Here we can see that we were able to capture mitochondria by looking at the presence of mitochondrial proteins, but we also see that there was some contamination from the peroxisome. In the antibody-based isolation method, the mitochondrial proteins were present, but there were also contamination from lysosome, ER, and the cytoplasm. From these Western blots, we can tell that antibody and the ultracentrifugation steps were better methods for isolating mitochondria. However, both methods have their own pros and cons, since ultracentrifugation, although it seems like it's better in comparison to antibody, it requires a lot of cells and it produces a very small yield of mitochondria. And the antibody method has some contamination from lysosome, cytoplasm, and the ER. So the future directions for my project would be to include more antibodies. So for example, we didn't have a nuclear antibody to test for the nuclear contamination, which would be essential if we're testing for RNAs, since there could be RNA contamination from the nucleus. And there is also another isolation method that we weren't able to try because of the limited time. It's known as mitoplast isolation, where mitochondria is treated with a detergent which bursts the outer membrane and the mitochondria and leaves us with the inner mitochondrial membrane. And with that, I would like to thank you all for attending. I would also like to thank the program coordinators, Sudha and Emma, my mentor, Sierra and Rebecca, and our PI, Seth, and all the members from the Shipman Lab for helping me during my time here. And I'd also like to thank Melanie Ott for the PUMAS grant and for this opportunity. Thank you.
Great job, Pemba. Any questions for Pemba? Feel free to unmute yourself and ask the question as well.